Hello friends, how are you? Have you completed your homework? Yes or no? Yes? Okay, very good. So, shall we start from this today's chapter? So, today's chapter is nutrition in animals. We already studied nutrition in plants, isn't it? So, in nutrition in plants, we already studied that plants are autotrophic. They prepare their own food material, isn't it? Now, we will start with nutrition in animals. And you know that animals cannot prepare their own food material. They have to depend on other organisms for the food. Therefore, that nutrition is known as heterotrophic nutrition, and the organisms, they are known as heterotrophs. But you know, different organisms having different types of food habits, and according to their food habits, they are having mainly three groups. First, herbivores, carnivores and omnivores. Now, what is the actual definition of that? Herbivores means those organisms which can take only plant as a food, and for example cow, sheep, buffalo, etc. After that, the second type, that is the carnivores. Those animals which take as an animal as a food, these animals are known as carnivores. For example, you know that tiger, lion, etc. While omnivores are those which takes plant as well as the animal as a food, these are known as omnivores. And for example, human beings, isn't it? So, on the basis of that, there are mainly three groups. Now, actually we have to study the nutrition. So, in that nutrition, actually 5 different steps are involved. And these 5 different steps, they are common in all the animal nutrition. So, first step of the nutrition is ingestion, second digestion, third absorption, fourth assimilation, and the last one, that is the egestion. So, the name itself indicates what is the meaning of that steps. Ingestion means taking the food inside the body, okay? After that, the next step, that is the digestion. You know, in the food, insoluble molecules are there, and the large insoluble molecules, they are converted into simple soluble molecules. But how they converted? That is in the process of digestion. In digestion, many enzymes are involved in these chemical reactions, and complex food is converted into simple food. Once the food is get digested, what happens? That food is get absorbed. That food is absorbed by the intestinal wall. You know, in the body, digestive system, and in that intestine is present. So, that intestine is helpful to absorb the food, and that food is entered into the blood. That process is absorption. After the absorption, assimilation. Assimilation means what? Simply, whatever the food is absorbed, enter into the blood. So, that absorbed food is reaches to each and every part of the body cell. And after that, what happens? Whatever the food is digested, some part is undigested. So, that undigested part is given outside the body, and that process is known as egestion. Egestion means removal of the undigested waste material from the body. Ingestion, the name itself indicates inside the body. Food is taken inside the body, and egestion means removal of the waste outside the body. So, now the next point is the nutrition in simple unicellular organism, that is amoeba. You know amoeba, that is single cell organism, and in that which type of food is taken? So amoeba takes the food, that is microscopic plants and the animals, and the mode of nutrition, that is the holozoic. And also the process of taking the food inside the body cell, that process is known as phagocytosis. The meaning of the phagocytosis is cell feeding. Okay. Now in nutrition, again same common steps like ingestion, digestion, absorption, assimilation and excretion. But the organism is unicellular. So there is no organ system is present. Simply single cell is there, and therefore diffusion is sufficient. Okay. Now you know how the food is entered inside the cell. Amoeba having no definite shape. This is the first thing. Okay. Amoeba produces a finger shaped projections, and these projections, they are known as pseudopodia or false feet. These pseudopodia, they are produces when amoeba comes in contact with food. These pseudopodia, they grows in a forward direction, and these pseudopodia come in contact with each other. Their contact will get dissolves, food and some amount of water enter inside the cell and forms food vacuole, and that is the first step, and that is ingestion. Now once the food is taken inside the cell, then what is the second step? What is the second? Yes, that is the digestion. Whatever the food is there, that food is get digested with the help of digestive enzymes. Inside the cell, lysosome is present, and you very well know about that. Lysosomes are known as digestive bags because they are having the digestive enzymes. So these digestive enzymes, they are entered inside the food vacuole. Food is get digested. So this process is known as digestion. Next step, that is the absorption. What happened in absorption? Digested food from food vacuole is directly get absorbed into the cytoplasm, and after the absorption there is no use of food vacuole, and that food vacuole becomes disappear. That's why the food vacuole is also known as temporary stomach. Whatever the food is enter inside the cytoplasm, it get assimilated throughout the cell for the energy as well as for the growth of the amoeba. And after that assimilation, what is the next and the last step? Yes, that is the egestion. But what happened, that they are having no fixed shape as well as no fixed opening to give the waste outside. That's why when considerable amount of waste is get collected, that time the plasma membrane or the cell membrane get ruptures at any point. Next example in that unicellular organism, that is the paramecium. Paramecium also having same type of nutrition, but only difference is that amoeba having no fixed shape, but paramecium having the fixed shape. It is sleeper shape, isn't it? And all over the body they having the hair like projections, and these projections are known as cilia. These cilia are helpful for movement of the food from outside to inside the body, and that opening from which the food is entered inside the paramecium, that is known as gullet. So these cilia, they can move, and because of this movement food enter inside the cell. Further processes, digestion, absorption, they are quite similar to that of amoeba. Now the next is the nutrition in complex multicellular organisms, that is example human beings. Means in short, we have to study the human digestive system. Human digestive system is mainly composed of digestive organs and the associated glands. But whenever you write this system, you write these organs in sequence. That is very, very important. So, digestive system starts from mouth. Mouth is an opening, and this opening opens into the cavity, and that cavity is known as buccal cavity. What is that? Buccal cavity. This is, okay. This buccal cavity and the nasal cavity is separated by hard and bony palate, and you know that this part, okay. This mouth opens into the cavity. In the cavity, salivary glands are present, teeth as well as tongue is there. So these teeth, they are helpful for chewing the food, for cutting the food, okay. The tongue is present. That tongue is helpful for mixing the saliva into the food. And you know, in the mouth, 3 salivary glands are present. These salivary glands secretes saliva, and inside the saliva an enzyme is present which is known as salivary amylase. Please remember, enzyme always ends with ASE. Don't make any spelling mistake. The food is taking inside the mouth. That food is carbohydrates. Carbohydrates means glucose and the starch, isn't it? So, salivary amylase act on the starch, and starch is converted into maltose by the action of enzyme salivary amylase. Is this clear? Okay. Now, this food remains only for few minutes in the mouth. Therefore, that food is partially digested. Now, this food is passed through from the esophagus. Esophagus is also known as food pipe. So, what is the use of that? Esophagus, no digestion takes place. Rhythmically contraction and relaxation of the muscles of the esophagus, that movement is known as peristaltic movement. So, this peristaltic movement is helpful for moving the food from esophagus to the stomach. Stomach is placed towards the left side of the abdomen, and it is J-shaped structure. Stomach wall having tubular gastric glands. These gastric glands secretes gastric juices, and this gastric juices contains 3 substances: hydrochloric acid, mucus, and protein digesting enzyme pepsin. And this pepsin act on proteins, and proteins are converted into peptones. But for this reaction, which reaction? For conversion of proteins into peptones, acidic medium is required, and this acidic medium is provided by, yes, exactly, by hydrochloric acid, isn't it? So, hydrochloric acid is helpful for providing acidic medium, okay. Now, the next substance which is present, that is the mucus. So, what is the role of mucus? Yes, mucus is helpful for giving the protection to the inner wall of the stomach. Inside the stomach, food remains for 3 hours, and it completely gets churned inside the stomach. Now, this food from the stomach is entered into the small intestine, and you know, in the stomach hydrochloric acid is there, means food is acidic, isn't it? Now, this food becomes alkaline in the small intestine. But how? That I explain you now, okay. Small intestine receives the secretions from liver and the pancreas. So, liver is the largest gland in the human body. It is about 2.5 kg, and it secretes yellowish green color substance that is known as bile. The spelling is B-I-L-E, okay. This bile is a combination of two substances, that is bile salts and bile pigments. Okay, this bile having mainly two functions. Remember, first function is the emulsification. What is the meaning of emulsification? Breaking down of fats. And second, bile provides alkaline medium, and this alkaline medium is required for the action of pancreatic juices. Now second associated gland, that is the pancreas. So pancreas is also large gland which is present beneath the stomach and parallel to the stomach. Okay. So pancreas secretes pancreatic juices. These pancreatic juices consist of enzymes like trypsin, pancreatic amylase as well as the lipase. Now pancreatic amylase acts on starch, and next that is enzyme lipase. Lipase acts on emulsified fats. So in this way small intestine receives the secretions from liver and the pancreas. So what happened? Liver secretes the bile, and that bile provides the alkaline medium for the pancreatic juices. So that food becomes alkaline in nature. Same the next time what happened? The wall of the intestine, they also having the glands, and these glands having different types of enzymes. Okay. So these enzymes, they completely convert the complex substances into simple substances. So for example, carbohydrates, that is complex, carbohydrate is converted into simple, that is glucose. So in this way in the intestine complete digestion takes place. What is the role of villi? That time you write this, villi, these are the finger shaped projections present inside the wall of the intestine, and these villi are helpful to increase the surface area as well as increases the rate of absorption. Okay. Now what happened? Intestinal wall absorbs the digested food, and that food is entered into the blood. Okay. And through the blood stream, this digested food, absorbed food is reaches to each and every part of the body cell. And the same, once the food is available, what happened? We get the energy. Now the next part is that whatever the food is digested, but some part is left as it is. It is not digested, and that undigested part is entered into the large intestine. Large intestine having two parts, colon and rectum. So undigested food from small intestine to enter into the large intestine, okay, in the colon first. Villi are helpful to absorb the water from undigested food, and because of that, once the water is get absorbed, that undigested food becomes solid, and that food is further entered into the rectum. Inside the rectum, this food gets stored for some time, and then it is released outside, or then it is removed outside from the body through the anus, and this exit of undigested waste is regulated by anal sphincter. So that process is last step of the nutrition, and this is egestion, removal of undigested waste. The length of small intestine is generally 6.5 meter long. It is completely get coiled in a compact space. But the length of the small intestine is quite varies with the animals. Sometimes question may be asked, why there is a difference between the length of small intestine? That time you have to write like this, because digestion of cellulose is quite difficult, and therefore they are having longer small intestine. but digestion of flesh or meat is quite easy, and therefore they are having shorter small intestine. We studied 3 different groups of animals like herbivores, carnivores, omnivores. Second point, in the nutrition, 5 different steps: ingestion, digestion, absorption, assimilation and egestion. Next point, unicellular organism, nutrition in amoeba and paramecium. And the last point, nutrition in complex multicellular organism, and that is the human beings. Means in short, we will study nutrition in amoeba and nutrition in human beings. Okay, nutrition in amoeba very simple, but nutrition in human beings, in that you have to concentrate. First, organs and the associated glands and related enzymes and how the process proceeds. So in this way you represent your answer and cover full marks. Thank you. Bye friends.